Hello students. So you all know that our chapter photosynthesis in higher plants is going on. In the earlier lecture we studied about the formation of ATP and NADPH. Now we'll study where are the ATP and NADPH used. And next is the primary acceptor of carbon dioxide and the Kelvin cycle. So we'll study about how and where ATP and NADPH is used. So ATP, NADPH and oxygen are the products of light reaction. Uh, Oxygen diffuses out of chloroplast while ATP and NADPH are used to drive the processes leading to the synthesis of food, that is sugars. It's called biosynthetic phase of photosynthesis. This process doesn't directly depend on presence of light but is dependent on the products of light reaction, that is ATP and NADPH, besides carbon dioxide and water. Melvin Kelvin, after World War II, worked with radioactive C14 and studied algal photosynthesis, which led to the discovery that the first carbon dioxide fixation product was a 3-carbon organic acid, that is 3-phosphoglyceric acid, PGA. He explained complete biosynthetic pathway and named it as a Kelvin cycle. Further work of scientists led to the discovery of another setup of plants in which the first stable product of carbon dioxide fixation was 4-carbon acid, that is oxaloacetic acid, commonly called OAA. So according to those plants in which the first product of carbon dioxide fixation is C3 acid, that is phosphoglyceric acid, they are called C3 pathways, and those in which the first product was C4 acid, that is oxaloacetic acid, they are called having C4 pathways. The process of photosynthesis starts when carbon dioxide is accepted by primary acceptor. The primary acceptor molecule is free carbon ketose sugar, and that is ribulose biphosphate. So the next step is in Kelvin cycle, which have 3 stages, that is carboxylation, reduction and then regeneration. So we'll study about the Kelvin cycle. Melvin Kelvin and his co-workers proposed Kelvin cycle. They showed that the pathway operated in a cyclic manner. The RUBP was regenerated, ribulose biphosphate. The Kelvin pathway occurs in all photosynthetic plants. It doesn't matter whether they have C3 or C4 pathways. So first step is carboxylation. Carboxylation is the fixation of carbon dioxide into a stable organic intermediate where carbon dioxide is used for the carboxylation of ribulose biphosphate. This reaction is catalyzed by the enzyme ribulose biphosphate carboxylase, which results in the formation of 2 molecules of 3-PGA. And the enzyme also has an oxygenation activity, so it's also called ribulose biphosphate carboxylase oxygenase or Rubisco. Rubisco is the most abundant protein present in the plants on the earth. So next step is reduction. Series of reactions that lead to the formation of glucose involves utilization of 2 molecules of ATP for phosphorylation and 2 of NADPH for reduction per carbon dioxide molecule fixed. The fixation of 6 molecules of carbon dioxide and 6 turns of the cycle are required for the removal of 1 molecule of glucose from the pathway. And the next third step is regeneration. Regeneration of the carbon dioxide acceptor molecule ribulose biphosphate is crucial if the cycle is to continue uninterruptedly. So this regeneration steps requires 1 ATP for phosphorylation to form ribulose biphosphate. For every carbon dioxide molecule entering the Kelvin cycle, 3 molecules of ATP and 2 of NADPH are required. This diagram shows the Kelvin cycle proceeds in 3 stages, you can see ribulose 1,5-biphosphate, that is RUBP, accepting carbon dioxide. And first step is called carboxylation, during which carbon dioxide combines with ribulose 1,5-biphosphate. And then second step is reduction, during which carbohydrate is formed at the expense of photochemically made ATP and NADPH. And the third step is regeneration, during which the carbon dioxide acceptor ribulose 1,5-biphosphate is formed again so that the cycle continues.